Friends, welcome to Biology Made Easy. Today in this video, we will discuss about centrosome and centriole. Centrosome and centriole. The centrioles were discovered by Baveri, and the meaning of centriole is cell center. That means the organelle which is present in the center of the cell, that means near the nucleus, is known as centrosome, is known as centriole. Now, what is the centrosome and centriole? Usually, when two cylindrical centrioles are present at a right angle to each other, that is known as centrosome or diplosome. That means now this is one cylindrical structure, and at the right angle there will be another cylindrical structure. And these two, they are surrounded by a proteinaceous amorphous matrix. This is a proteinaceous amorphous matrix. That means both these centrioles, they are suspended in an amorphous matrix, and this matrix is known as centrosphere. This is known as centrosphere or gynosoplasm, or also known as cytocentrum. So both these together, it is known as centrosome, or also it is also known as diplosome. And each one is a centriole. Now, what is the structure of the centriole? Usually, the centriole is membraneless. That means it is not surrounded by any membrane. It looks like a cartwheel. And around this centriole, other organelles are not there. That means the cytoplasm around is completely free. So it has a zone of exclusion. It is known as zone of exclusion. So what is the structure of this each centriole? Each centriole usually consists of 9 peripheral triplets. So likewise, there are 9 peripheral triplets, and they are designated as A, B, C. That means these 3 are known as peripheral triplets, and each one is a microtubule. While A has 13 protofilaments, B and C, they have either 10 or 11 protofilaments, because they are fused with this. Now, all these triplets, they are made up of protein tubulin. Now, again, the C of one peripheral triplet is connected with the A of another by means of linkers. Likewise, these are the linkers. So these are the linkers. and at the center there is a hub. This is the hub, and to this hub usually they are connected by means of radial spokes. The A of each triplet is connected by means of radial spokes. So these are the radial spoke. So you see, it is completely membraneless. The cytoplasm around is completely that is free. That is known as zone of exclusion. Now it has 9 triplet microtubules. That will say 9 triplet microtubules means 9 into 3. That means 27 microtubules are there. Microtubules, and all of them are made of a protein tubulin. And they are tilted at an angle of 45 degree. 40 to 45 degree, they are twisted towards the axis. So the linkers and the spokes, they are also made up of the same protein tubulin. That means in the centriole there is only one type of protein, that is only tubulin is there. And this centriole, it is present in eukaryotic cells, and mostly it is present in case of all animals and lower plants like algae, fungi, and bryophytes and pteridophytes. Bryophytes and pteridophytes. These are not present in case of higher plants. In higher plants they are completely absent. So in these cases, what happens, that means the centriole has mainly 2 functions. One important function is the centriole is usually present at the base of the cilia and flagella. It is present at the base of cilia and flagella. So for what purpose? For the purpose of locomotion. So this is its primary function. That means it helps in locomotion. That means wherever there is a cilia and flagella, there must be centriole at the base, which is known as the basal body. But wherever there is centriole, there may not be cilia and flagella. So this is the main function. Another function is in case of animal cells. In case of animal cells, it helps in formation of helps in cell division, because it gives rise to the spindle fibers. It gives rise to the spindle fibers. But the cell division is secondary function, while the locomotion is the primary function. You may ask why. The reason is, in case of animal cells, since it is present, it helps in formation of spindle fibers. But in plants, there is no centriole. Then how the cell division takes place? That means without the centrioles, the spindle fibers can be formed. That is why the locomotion is primary function and cell division is secondary function. And during cell division, when there is cell cycle, the centrioles replicate at G2 phase. At G2 phase, they replicate. That means each centriole becomes 2, so that they can move to either poles. But very important thing, that means the centriole has capacity of replication even in absence of DNA. It replicates without DNA. And how it gives rise to the spindle fibers? Usually in the peripheral part of this centriole, we will get some structures like this. These structures are known as maculae. These structures are known as maculae, or they are also known as pericentriolar satellite. Pericentriolar satellite, or they are known as microtubule organizer center, in short MTOC, microtubule organizer center. That means these masculine or pericentrial satellites, they are the centers of formation of these microtubules, which are required for formation of spindle fibers. So this is the structure of the centrosome and centriole, and it is membraneless. It looks like a cartwheel. It has a zone of exclusion. It has a capacity of replication of G2 phase even without DNA. And it has mainly two functions. One is help in locomotion and the other is cell division. But locomotion is the primary function while cell division is the secondary function. And the entire centriole consists of only one type of protein, that is tubulin. So now you have a very clear understanding about this centrosome and centriole. It is present only in animal cells, absent in plant cells. So thank you very much for watching the video. Wish you a very bright future. God bless you.